Hi guys, Dr. M here with a video that's going to take you step by step through annotation of a Drosophila elegans gene on the 4th or dot chromosome. So we're going to start out going to gander.wistl.edu and we'll go to the genome browser. And I've got everything all set up already, but you've done this before. And I'm going to stick with Contig 25, which I randomly claimed, and so this is what it looks like. So the first thing that we're going to do is take a look at this one of these genes. It doesn't really matter which one. So I'm going to start with this BT. So I just clicked on the name of it, and I'm now going to click on the flybase ID. This is going to take me to the flybase protein entry for this, and it's going to give me the protein sequence. Sometimes it's going to look like this when you first get there, but if you just hit the sequence, it'll come up. And so you're going to copy and paste this. This is fairly long, and I did do this in class last time. And then we're going to, I'll open a new window and go to the flybase website and go to BLAST. I'm going to stick with genome assembly, but I'm going to switch this to tBLASTn. So we're going to go from amino acid to the nucleotide sequence. So last time, why this wasn't working is because we were trying to do amino acid to amino acid, BLASTp. That is not going to work because Drosophila elegans doesn't have any annotated proteins. So that's kind of what our job is. And so we have to do the tBLASTn for this. So that's why it wasn't coming up with anything significant, no significant hits. And if we set it up and then pick our, so set it up with the genome assembly and the tBLASTn and then paste it in. We're going to go down here, unclick Drosophila melanogaster and click Drosophila elegans. So what we're doing again is we are blasting the Drosophila melanogaster protein amino acid sequence for this gene, and we're blasting it against Drosophila elegans assembly. So I'm going to hit blast. And this is what we get. The blast search was not actually that fast. I just paused it and skipped ahead. That took probably a couple minutes to run. So I'm going to note the length of this query. This was, I guess, that many nucleotides, 8,900 nucleotides long. And what we're looking at here is there are regions in this area that have a fairly strong conservation, fairly strong homology between melanogaster and elegans. These areas are less strong, less conserved. And so what we're going to do is, what I'm doing here with this search is I'm going to get the coordinates of the protein in the Drosophila elegans genome. And so in order to do that, I want to get the entire protein. And so I'm going to start, instead of starting with this first hit, which is maybe arguably the strongest, instead I'm going to start with the hit that lines up with the very first nucleotide, and then I'm going to look at the hit that lines up with the last one. And so these are not bad hits. I mean, they're not red, but they're the next best thing. And so I'm just going to click on this, and it'll take me directly to it. And actually, that's starting at query 8. I would rather start at query 1, so I'm just going to get back up there and try this one, which, query 3. Okay, so we're not ever really quite there, are we? No, that may be as good as it gets. But okay, I'll try this one just for kicks and see what happens. Oh, we're at 1. Okay, so here, I'm going to write this down. So we're going to note this starting coordinate, because this is our position in the Drosophila elegans genome, or on that chromosome, on the dot chromosome. And we are then, I'm just curious, I just want to know if this coordinate, so this is 5191130, and I just want to know if this hit is in a similar place, and that's actually in a totally different place, and that's 3441883, and that's good to know. 3441883, and I'll just click on a couple others and just see if these are, that's in the same place, so that's 3441526, and I just want to click up here and see who this belongs to. So this belongs to the 439983, which is perhaps even still different. So I'm going to write that down too. Okay. And so these are kind of looking like they're all over the place. So that's good to know. So that's the front end. Let me just check this one too. So that's 429, so that's the same place as the last red one, which was 439983. Okay, so that's consistent. All right, let's look at the back end. Let's see what this one is looking like. So now instead of looking up here, we're gonna look down here because this is our last one. And I forgot to check. So this is close to the end of our query. It's not exactly at the end, because I think it was 8933. So this was 85, our last is 850186, which seems quite different from any of the other ones that we saw. And let me just try this one. And so that one is 5192374. Okay. Which does match one of our beginning coordinates, and of course I can't remember which one, but that's okay. So we have a fair idea of what's going on with this. We're getting multiple hits on multiple places in the elegans genome, on the elegans chromosome, and we don't. These are scaffold names. We don't actually know what chromosome they belong to, but they are all different. So on this scaffold, these are all hits. Okay, so our next step is going to be, I also want to take a look at this other gene. I'm going to go back to my genome browser and I want to look at this. And maybe actually before I do that, I would like to know where my Contig sits in the elegans genome. So I can compare and say, okay, we have all of these coordinates. Do any of them actually fall within where the Contig ends up? And so here's where we're going to blast the beginning and the end of the Contig. And so we're gonna, whoops, not here. I'm gonna go back. I'll have to go back a little ways. All right, so back to my blast. I'm gonna clear this, and I am going to go to my Contig folder, to the source file. We've got my FASTA here. I'm gonna open that up. And I'm just going to go down maybe this far. I'm going to copy and paste that here, and I'm going to blast it. I still have elegans picked. So now we're blasting the beginning of the elegans Contig, Contig 25, to the elegans assembly, the whole genome, just so I know what coordinates show up for that. So we're going to do this and hit blast. Okay, so I made a mistake. I got a warning because I had T-BLAST-N chosen to go from amino acid to nucleotide, but I actually have nucleotides here. So I'm going to switch this from nucleotide to nucleotide to nucleotide to the blast N. I'm going to try that again. Okay, so I got a really nice, strong single hit. All of the rest of this is meaningless and not significant because we're in the very low color range for the scores for that search. So I'm going to click on this, and now we're at the beginning of our query. Our position for this is in the 469997. That's the beginning of my Contig. And then I can kind of look and see, okay, does this actually line up with any of the coordinates of my previous gene that I blasted? All right, so and I think it might, but we still need the end of it. So I am, whoops, there it is, okay. So I'm going to just scroll down to the end of my Contig, and I'm gonna take, say, that many, and I'm gonna go back here and blast it again. All right, so I'm gonna clear this, throw that in there, and do this again. Okay, awesome. So we got another nice, pretty strong hit that, and then a whole bunch of not very good hits. And so this is what we like to see. So this is 228.50 in length, and I want to make sure that this hits the last nucleotide, which it does, and that ends at 411998. Now, you may note that this coordinate is smaller than our beginning coordinate. That is okay. That just means that our orientation of our Contig is just reversed, and you'll see that in this plus minus indication, and that's fine. That is totally okay. We don't need to worry about that. But when we are looking at where our gene falls, we do still want it to be falling. And we're going to find that when we're blasting our genes against the elegans assembly, we're going to also be seeing the beginning coordinates of our gene that are larger than our ending coordinates. And so they should also be in a plus minus orientation with the genome assembly, because the genes that we're getting from the Contig should match the orientation of the Contig. It's okay if both of them are in the opposite orientation of the assembly. Okay, and so now we have our beginning and ending Contig, and we can go back and look at, so we had one coordinate from that past search, which was 439983. That does fall within here, so it is possible that this BT gene might be actually where we think it is. However, what we need to do is blast this one also. So this SLS, I just clicked on it, and we're going to click on the Facebook, I did it again, the flybase entry, and we're going to copy and paste this guy. It's also very long. All right, and we're going to, I'll close this, and we're going to blast it against the genome assembly, and this time we're going to do T last N again, okay? So be sure to switch that around. All right. All right, let's look at this. So, all right, so let's see if we can do the same thing and find, I'm just going to click on this and see where it ends up. So that is 3385029. We're on query 60, on the 63rd position, which is not quite hitting where we think it is, and this is outside of our Contig. So let's take a look. I just want to take a look here. So that's 338. 88678, also